and I will be moderating today. Today's topic is automated 3D cell culture and screening by imaging and flow cytometry. We will be recording the session and will make it available on our website along with the PDF of the slides within 24 hours. During the session, you can type questions into the question box on your screen. After the formal presentation concludes, we will have a Q&A period and address as many questions as possible within the time frame. We will respond by email to the questions we do not have time to answer directly. Our presenters today are Dr. Nikki Slani, Applications Director at 3D Biomatrix, and Dr. Mike Kowalski, Staff Application Scientist, Beckman Culture. Nikki has over 15 years of tissue culture and molecular biology experience. She received her PhD from the University of Michigan, where she trained as a developmental biologist with an interest in the mechanisms of stem cell differentiation. Mike Kowalski is a staff application scientist in the sample preparation and applied markets groups of Beckman Culture, Life Sciences. He received his PhD in microbiology and molecular genetics from Harvard University. Prior to joining Beckman, Mike was a postdoctoral fellow at the Novartis Institutes for Biomedical Research, where he used automation to screen for novel regulators of stem cell pluripotency and differentiation. Since joining Beckman, Mike has developed automated applications in the area of cell culture, cellular analysis, and mass spectrometry. Nikki will start the session by providing a brief overview of the hanging drop plate technology, and then we'll turn the session over to Mike. Go ahead, Nikki.
Thank you, Marianne, for that very kind introduction. Today, as Marianne mentioned, I'll be covering why you might want to culture cells in 3D, and then creating and feeding 3D cell cultures in the Perfecta 3D hanging drop plate. So this slide illustrates an example of a 3D culture on the left, and for this example we've chosen a hanging drop plate, versus a 2D culture on the right in the more standard format that people are used to seeing, being either a well plate or maybe a round bottom dish. So what you notice when you look at these two different ways of culturing cells is on the left, in a 3D cell culture, physiologic cell-to-cell contact dominates. And cells are able to interact with extracellular matrix in a much more physiologic way. And these two things create diffusion gradients of drugs, oxygen, nutrients, and waste, which I'll illustrate a little bit more on the next slide. And if you were to use co-cultures of multiple cell types, you can actually mimic microenvironments found within normal physiology. However, if you compare that to 2D culture on the right, you see that cells in 2D culture are mostly in contact only on their edges. And they're touching really, the vast majority of what they're in contact with is just plastic. And they can contact extracellular matrix, but mostly typically on one surface, maybe two surfaces. And because of this, there's no real gradients present. All the cells in that culture see the same oxygen tension, they see the same amounts of nutrients, they see the same amounts of waste. So you have none of these gradients present. So if you make a co-culture, it's really unable to establish a typical microenvironment. So here's a slide that compares spheroid on the right as a microtumor model illustrated on the left and what it might look like in vivo. So what you see here in vivo on the left are the quiescent cells around the outside of a the proliferating cells around the outside of a microtumor, which are growing because they have exposure to oxygen, nutrients, as illustrated on the arrows on the bottom, in comparison to the cells more deeply found within this microtumor that actually become quiescent, mostly due to a lack of access to oxygen and a lack of access to nutrients. And if these microtumors become large enough, you can actually end up with necrotic cells in the center because they're unable to access oxygen and nutrients, and they're unable to get rid of CO2 and waste. And this is all a function, again, of this 3D structure and its interaction with blood vessels in vivo. So you compare this to microtumors models in vitro, a spheroid. These cultures are 100 to 800 microns in diameter. They're a very simple, well-established representation of these microtumors. They're very easy to form, and they have all these really important diffusion gradients that really are critical for the physiologic interaction of cells with drugs or just trying to understand different model systems. So this slide illustrates Perfecta 3D hanging drop plates. They're available in standard 96 well and 384 well formats. They're made of polystyrene. They're packaged individually and sterilely, and they're compatible with common manual and automated equipment. And Mike will do a really excellent job of discussing some automation of these plates a little bit. So on the bottom you see a cross-section through the hanging drop plate. It's comprised of 3 pieces: a bottom tray with a reservoir to provide humidity, the hanging drop plate where the drops actually attach, also with another reservoir to provide humidity, and then a lid to keep the entire assembly sterile. And if you look in the upper right-hand corner, what you're seeing is a view from the bottom of the plate, and you see the drops hanging below that hanging drop plate. So this slide illustrates how you would first seed cells into the hanging drop plate and then feed the cells. But I would like to point out this is illustrating with manual methods. And so you see the pipettes on the angle. That is not necessary to load the plate, and with automation it's very easy to load. Again, that will be discussed a little later in this webinar. So across the top you see you dispense the cell suspension into the well about 3/4 of the way down, and the surface tension of the media forms a hanging drop, and then the cells are driven by gravity to the bottom of the hanging drop, where they aggregate, and then given enough time, between 1 to 5 days depending on your cell type, they form a spheroid. And what you see across this top line of cartoons is there's no contact with plastic during cell culture. And then on the bottom, this cartoon illustrates how easy it is to feed cells or add reagents into the hanging drop plate. Because the spheroid is at the bottom of the hanging drop, it's very easy to use a pipette tip to aspirate media from the top and then dispense fresh media into the top without disturbing cells in the hanging drop plate at all. This slide illustrates across the top that you get precise control of spheroid size with Perfecta 3D hanging drop plates. So this graph shows spheroid volume versus days in culture for a variety of different seeding types, and what you can see is that they're very tightly grouped. So you can make small spheroids by seeding small numbers of cells and larger spheroids by seeding large numbers of cells. The cartoon across the bottom illustrates that feeding cells maintains osmolality of tissue culture media. As these are hanging drops, you might be concerned that there would be evaporation, which could change things like osmolality, but simply removing some percentage of the media, which depends on your incubator and its humidity and its ability to maintain humidity, and then adding media back roughly every other day or every 2 days, again depending on your cell's physiologic needs and the humidity conditions of your incubator, was shown to maintain the osmolality of media in 3 different cell types over approximately 12 days of cell culture. So you don't have to worry about changing osmolality being toxic to your cells. So this slide illustrates the flexibility of the hanging drop plates and the ability to optimize spheroid formation. About 50% of cancer cell lines spontaneously form spheroids. And the top set of figures is from a paper where the authors determined some of the innate cellular properties that influence spheroid formation. So what you see, there are 3 types of hepatocellular carcinoma cells, and they really demonstrate the different types of spheroids that are naturally formed by cells. The Hep3b form a very tight spheroid, Hep-G2 form aggregates, and the PLC cells really don't form spheroids at all. And what the authors discovered is that beta-1 integrin expression was relatively high and close to equal, but that ECadherin expression changed between those cell types. And I don't want to go into too much detail about their experiments, but it's an excellent paper. And that paper allowed those authors to create the bottom cartoon, which illustrates the model the authors developed based on this data. And that cells first aggregate based on integrin and ECM interactions, and then there's a delay period as they accumulate ECadherin, and that causes the eventual spheroid compaction. And that's another really excellent paper that helps you understand the theories behind spheroid formation. So a big advantage for the hanging drop plates is to be able to add media-soluble extracellular matrix, for example 2.5% Matrigel, or other matrix components, or packing agents such as methylcellulose or perhaps polyvinyl alcohol, either at the time of plating cells or a few hours later, to alter spheroid formation and timing and compactness to suit your experimental timeline. So you can use this to set up a model that closely mimics the physiology that you're trying to generate. So another strong characteristic of the hanging drop plates is that you're able to create very powerful co-culture methods. For example, the first line across the top illustrates the one-time seeding of multiple cell types. In this case, the example you see is red fluorescent protein expressing cells seeded at the same time as non-red fluorescent protein expressing cells. And what you see is the cells then self-pattern into sort of what appears to be a randomly organized spheroid, but the cells will have some role in self-patterning the spheroid. Then across the next line down, you see sequential seeding of multiple cell types. In this case, it is green HEK cells being seeded to form a spheroid, followed by red HEK cells that are used to coat the spheroid. So you can do sequential seeding of multiple cell types to create a pattern spheroid. Again, being able to make the kind of model system you're trying to replicate. And finally, you see simultaneous culture of multiple cell types, for example, red cells and green cells, both used to form spheroids. And then because you can remove the spheroids through the top of the hanging drop plate, you can actually put two spheroids into one hanging drop together, allow them to make contact and bind with each other, maybe invade each other. And the authors of this paper named these Janus spheroids. And it's another interesting methodology to look at perhaps invasion or cellular-cellular interactions. So again, the hanging drop plates allow you very flexible platform in which to create these really powerful co-culture methods to make the cell model of your choice. So how can you analyze 3D spheroids? Basically the same way you analyze 2D cultures. And again, Mike is going to do a great job of really addressing some of the constraints and important points about doing this. But essentially you would seed your cells in the hanging drop plates, allow spheroids to form, add test compounds, and then perform quantitative assays and qualitative assays that you're used to performing. So you can perform spectrophotometric assays, you can do staining with immunohistochemistry or cytoplasmic staining. You can embed them and section them. And very interestingly, they can even be analyzed by flow cytometry as well as microscopy. So we have lots of protocols on our website, and we're always willing to offer help as much as we can. So with that, here's my contact information. If you have any questions about using the hanging drop plates, I'd be happy to talk to you anytime. And with that, I will say thanks and turn it back over to Marianne.
Thank you, Nikki. Now we're going to turn the presentation over to Mike, who will be talking about his work on automating the process for screening and flow cytometry. Go ahead, Mike.
Thanks, Marianne, and thanks to Nikki for the introduction into the perfected 3D plates and 3D culture in general. Also, if there are questions that we're unable to get to today, my information is shown here. It's mkowalski@beckman.com. I'd be happy to try and answer any questions that I'm able. So just a brief overview of what I'll be describing today. I'll just touch briefly on reasons why people seek to automate their various applications, and in particular how these advantages can then be applied to 3D culture. I'll show some representations of how we've been able to automate the 3D culture on the perfected 3D plates, as well as compound treatment and two different forms of screening using what we consider to be very complementary analyses, which, as previously mentioned, are high content imaging and flow cytometry. And then I'll just briefly touch on how one then scales up to automate a full screen. So reasons as to why people would seek to automate their application. One is frequently repeatability. So this can apply to both accuracy and precision, and basically automation can help improve on what we're able to do manually, particularly if you're seeing a significant user-to-user variability, so when multiple people in the lab are trying to perform the same experiments. Also, automation can help reduce the opportunity for errors. And in addition, it can free up resources. For any of us who have spent significant time at the bench, we know there are particularly slow or complex workflows that can drain a significant amount of our time. Or even simple assays, if done at a high enough throughput, can again require an awful lot of bench time. And these are applications that automation can make a real significant contribution towards. One consideration when thinking about automation is whether it's preferable to have automation that is dedicated to a fixed purpose, so essentially just doing one assay, but typically is very easy to use, versus having a platform that's more flexible and can be used for multiple applications, but might require a little more effort to get there. So how do these benefits apply to 3D cell culture or the use of perfecta plates? Well, when it comes to repeatability, the automation is able to give very fine control, both whether you're discussing pipette location or the speed at which liquids are pipette. And this can be done consistently time and time again. So again, really able to repeatably deliver the cells and culture them on these perfected 3D plates. With regards to freeing up resources, obviously these are not just your straightforward 2D culture where you can just throw media into a well. So that it does require a bit more manual effort, and certainly easier to do it, say, 96 channels at a time than a single channel at a time. And also, because you're forming these 3D spheroids, these are multi-day cultures. So again, these are plates you're going to be coming back to day in and day out for, again, up to anywhere from 5 to 12 days perhaps. So again, significant effort is required. And another consideration when it comes to automating any cell culture, including 3D, is how to maintain sterility of your cultures. And for this, we provide sterile tips, as well as the option to enclose the automation within a HEPA-filtered enclosure, which basically acts like a laminar flow hood. So a brief introduction into the automation I'll be talking about. At Beckman Coulter, we make the Biomek Workstations, and they come in 3 different versions. There's the Biomek 4000, which can alternate between single and 8-channel fixed pipetters. There's also the Biomek NXB, which has a single head that can either be a span 8-channel, where your 8 channels can spread out from one another, allowing you to access multiple tubes at once. Or it can have a multi-channel, either a 96 or a 384 well head. But however, the work I'll be showing today was actually performed on the Biomek FXP, shown to the right. And this actually has 2 heads. In our case, it has one span 8 and one 96-channel multi-channel head on the left. All 3 platforms share the same intuitive software. Basically, an automation method is created by just dragging and dropping steps, whether it's an aspirate or dispense step or a moving labware step. And then each of those steps can be configured either just at a very general level, where you would show a source and a destination for a transfer and what volume needs to be transferred, or you can have finer control when needed, such as where within the well you want the pipette tip to be located, underneath the liquid or at the very top of the well, for example. And also, data is tracked throughout an automated method. So for example, if a bar-coded plate has a variety of known samples in them, and those samples are transferred to another plate, all the data associated with those samples are now found in the new plate. So again, nothing should be lost during an automated method. In addition, I spoke of flexibility earlier, and the Biomeks are inherently flexible. First, you can configure the deck in any way you see fit, and in addition, you can integrate additional items such as positions that can be used to heat or cool plates or samples, that can shake plates or tilt plates if you need to aspirate from the side of a well. And you can also integrate additional instruments such as various analyzers, whether those are plate readers, flow cytometers, high content imagers, or you can integrate incubators for these long-term assays that I'll be describing today, barcode readers, plate washers, etc. So again, very flexible instruments so that it can really accommodate a large variety of workflows. This is an image of the cell culture system we've set up in the lab. Again, as I mentioned before, the Biomek FXP is shown in the center of the HEPA-filtered enclosure. And basically, again, this can just pull filtered air onto the surface of the instrument so that reservoirs that are open or when plates are delidded, they're maintained in a sterile environment. In addition, for a full-blown screen, you'll need additional storage shown on the left, which can be used to hold plates or additional tip boxes that are then brought onto the instrument using the shuttle and the barcode reader shown adjacent. On the right-hand side is a CO2 incubator that we were using to culture the cells. Again, plates are brought back and forth from that item as well using a shuttle. And in the front is the ViCell XR cell viability analyzer that uses trypan blue staining to basically quantify the percentage of cells in a solution that are viable. So what we wanted to use this system for was basically to execute a compound screen where we wanted to automate the plating, the culture, and the eventual compound dilution and treatment of the hanging drop spheroids that we made from HCT116 colon cancer cells. And in addition, at the final day 4 or day 5, we would then process these spheroids for analysis of apoptosis. And so again, shown below is the workflow where we would plate cells on day 0, treat them with diluted compounds on day 3, and then do the sample prep and analysis on day 4 or day 5. And all the data I'll be showing today is from day 4. So we'll go into each of these days in a bit more detail and show how we were able to automate these processes. So when it comes to cell plating, day 0, we found with our just kind of manual optimization that adding 4,000 of the HCT116 cells and 40 microliter droplets on the perfecta plates would eventually give us a nice spheroid. But what we did find is that by using 0.25% polyvinyl alcohol, which Nikki alluded to earlier, that we were basically able to accelerate the aggregation phase of the spheroid formation, and that's shown on the images on the right, where after 24 hours of culture, the upper image shows, again, significant gaps between the cells that are still kind of drifting apart, but in the presence of PVA, the cells have clustered into a single cluster at the bottom of the droplet, and that basically resulted in us getting very robust spheroid formation by day 3. So to automate this process, we set out certain goals. We obviously wanted to get consistent cell distribution across the plate so that all our spheres would be equivalent so we could test them equally. We wanted to make sure that we did not lose any drops in the pipetting of the cells, and we wanted to avoid contamination. And obviously that's the case for all 4 of the steps, but I'll just mention it now that we did culture the cells in the absence of antibiotics and never detected any contamination, indicating that the sterile environment and the sterile tips were sufficient to prevent contamination. So here I'll actually show a brief video that describes and illustrates the automation. It begins with just delidding the perfected 3D plate. And shown here is the 96 multi-channel head just taking a single column of tips using what's called our enhanced selective tip pipetting. And this allows us to access a smaller reservoir of cell suspension, and we basically mixed the cells to make sure we had an even distribution, and then pipetted them into the perfected 3D plate, again using our very thin P50 tips. You'll notice this extra push down. That basically helps the droplets hang underneath the plate, as Nikki described earlier. And that'll be illustrated again here in slightly more detail. So again, dispense the liquid at the level of the rings and then basically gently push them through. And that was sufficient to give us nice consistent plating. The plate is then relidded. And one of the nice things here is we were actually able to control the speed at which plates are moved in a plate-specific manner. So the system knows anytime it picks up the perfecta plate, it should move very slowly so as to not disrupt the hanging drops. The plate is then sent on the shuttle to the incubator, passing through a barcode reader, and then incubated for 3 days. So the results of this stage is that we were able to process a full plate using this technique in less than 8 minutes. We did find that we were able to achieve consistent hanging drops, and that was due in large part to the plate definition and pipetting techniques that were provided on the 3D Biomatrix website. As Nikki alluded to, there's a vast amount of resources for working with these plates, including how to use them proficiently with the Biomek FXP. I also mentioned how we slowed down the plate movements to ensure that the drops weren't lost due to a jarring motion. And I mentioned how we use the enhanced selective tip pipetting to not only were we able to pipette to partial plates, which was great for the initial optimization type areas, but we were also able to significantly reduce the dead volume that would be required to plate these cells. So even plating in a full plate, if you were using 96 channels at a time, a reservoir that can hold the entire 96 tips requires a dead volume of over 20 mils, versus just using a single column at a time. We were able to get that dead volume below 1 mil. So again, significant waste lessening there. When it comes to consistency, we wanted to, again, make sure that our spheres were consistent across the plate. So we actually used our imager to look at size and circularity. And shown to the right are 48 wells of hanging drops with day 3 spheres shown. And basically what you can see is across the plate, although one well was lacking a droplet, we did find that occasionally well D7 on a small portion of the plates would not retain liquid, whether that was through automated or manual fashion. But for the remaining 47 spheroids, we analyzed the area and perimeter, and just looking at the CVs, which were below 6%, gives a nice indication that the spheres were consistent across the plate. In addition, we looked at a shape factor, which is a measure of circularity. And a perfect circle is basically has a shape factor of 1. So to get a shape factor of 0.85 really indicates you had an excellent sphere. And again, a very tight CV, so we had a nice consistency across the plate. So after 3 days of incubation in our adjacent incubator, it was time to treat them with a variety of compounds to try and induce apoptosis. And what this entailed from an automation perspective was to basically we placed 100 microliters of our diluted compound in the first row of a plate. And this would be anywhere from 2 to 6 compounds depending on the experiment. And we would do 2 columns each, mainly because we wanted to do 2 different analyses for each condition. We then did a serial dilution in a row-wise fashion at a 1:2 ratio down to the second to last row, and then the last row we just had our DMSO only control. We then replaced 10 microliters of the media in the droplet with the compound containing media and basically incubated those cells for 24 hours. Again, our automation goals here was to have an accurate serial dilution, to be able to remove the media without disturbing the spheroid, as Nikki demonstrated earlier, and to again ensure that we had no lost drops due to either movement or pipetting. And again, this will be a brief video showing the automation that was occurring. So in the background, you can see our span 8 pipetters that I mentioned earlier, basically accessing the reservoir that has DMSO containing media for our dilution curves of the compounds, and basically dispensing it into the 7 wells that do not already contain compound. Same thing, just shown from the front, again accessing the bottom 7 rows of our compound plate. And then the next step, we again use the enhanced selective tip to basically just pick up the 3 tips we needed to do a row-wise dilution curve. And again, moving row-wise is something that's not necessarily easily done with a span 8 pipetters or fixed 8 pipetters. So what you can see here is again aspirating the compound, moving 1 row further up, dispensing and mixing, and then again aspirating that well and moving to the next row, again for part of the serial dilution. Following this, the HD plate is delidded, and we picked up 24 tips in this case, and basically removed the 10 microliters from the top of the droplet, dispense that to waste, and then pick up the 10 microliters of diluted compound, add that to the HD wells, and then basically just do a gentle mix to help distribute the compound. After this, the plate was relidded and returned to the incubator for another 24 hours. So the results of this particular step was that we were able to process a full plate, including the serial dilutions, in less than 7 minutes. We were able to actually observe that there was a visible dilution taking place because some of the compounds we used were actually fluorescent, and in addition we could actually see some effects on the spheres in a dilution-wise fashion. And again, this dilution was facilitated by the enhanced selective tip pipetting that I mentioned earlier, which allowed us to do this in a row-wise fashion. No droplets were lost during the pipetting, and this actually was a slight challenge because due to the kind of more inherent resistance of these 3D cultures to these apoptosis inducers, we had to start at fairly high concentrations, and that translated into fairly high concentration or high amounts of DMSO in the media. And what we found is that this amount of DMSO was sufficient to change the surface tension, and the droplets became a bit more unstable. But because we used the media removal step previously, the final 40 microliters in the presence of DMSO was still sufficient to keep the drops in the hanging drop fashion. In addition, we did not lose any spheres during the pipetting step, again due to the fine pipetting control that we're able to achieve with the automation. So after 24 hours of incubation, we then had to do 2 different forms of sample prep and analysis because, again, we wanted to basically show how high-content imaging and flow cytometry are both excellent ways of analyzing cell cultures, but both have challenges when it comes to analyzing 3D cultures. And it turns out the 2 are very complementary, and hopefully I'll be able to describe that. So shown here is basically just a little more information about high-content imaging. For those who aren't familiar with it, what it is essentially an automated microscope. And for all the data we generated today, including the previously shown 48 wells in the bright field hanging drops, we use the Image Express Micro from Molecular Devices. And what this does when normally working with a 2D adherent culture is gives you phenomenal cellular or even subcellular data where you can take high magnification images of cells and even determine if your fluorescent markers are cytoplasmic or nuclear or even on the cell membrane. And the way you can make this more quantitative is to take multiple images per well until you've basically imaged enough cells that you can get quantitative data. However, in a 3D culture, the challenge is that there's not a single plane of focus. Obviously, this is a 3D structure, and in addition, it's hanging in a droplet. So to try and get this sort of level of detail and basically get cellular data is very challenging. In addition, if you wanted to try and use a higher magnification but take different images at different locations in the sphere, that's exceptionally challenging because the sphere is obviously not in a fixed location. It is able to essentially move around within the droplet. So even if you took multiple images, they're probably going to end up being in different planes of focus. So what this translates to is that basically you end up requiring a low magnification to take an image of the entire sphere. However, one of the nice features of the Image Express is the newest XL system has a wider field of view, which enabled us to take these images of the spheres at 10x magnification. In contrast, flow cytometry is typically used to analyze cell suspensions, and for the data we generated today, we used the Beckman culture Gallios. So because this is best suited for suspension cultures, or essentially requires suspension cultures, people typically use it for either blood samples or other 2D cultures that you can then get into a suspension by trypsinizing the cells to basically get them into suspension. And the nice thing about this is that you can get cellular data as the cells pass in sort of a single file through a laser, and you can get cell size and cell fluorescence, depending on whatever markers you've stained for, even upwards of multiplexing 10 different fluorescence colors. So you can get thousands of data points per sample just looking at one cell at a time in a very short time. The data basically looks like the images shown down below, where if you're looking at a single parameter, people frequently show it in a histogram format as sort of a positive-negative population. Or if you're looking at multiple parameters, you might see it in a dot plot fashion, where you're plotting two different characteristics, such as green and red staining. Everything we'll be looking at today will look like the histograms. We'll really just focus on staining in one color at a time. So again, this is great for cell suspensions, but for 3D culture, obviously this presents a challenge because you need to dissociate the spheres into a single cell suspension. And what this usually entails is adding some sort of enzyme such as trypsin or collagenase to essentially chew up the extracellular matrix, and then repeatedly use mechanical disruption to try and break up the sphere into these suspensions. Another drawback is obviously that this becomes an endpoint assay. Obviously your sphere is no longer a sphere, and in addition the cells that you run on the flow cytometer are actually sent to waste unless you happen to have a cell sorter. So one of the advantages to imaging might be that you could look at multiple time, or essentially use a time course and watch your sphere over time, where flow cytometry again is an endpoint assay. So I mentioned that we feel these two analyses are complementary, and I'll just give you one example where this might be the case. So shown on the left is basically one of our images that we had of a sphere that was stained for cell nuclei using a Hoechst 33342 dye. So that's the blue color. And then the sphere was also stained with annexin 5 as a marker of apoptosis. And what you can see is it appears that only a small portion of the cells are staining positive for annexin 5, and all of them seem to be located on the outside of the sphere. Now this could actually have two interpretations. This could mean that only the exterior cells are undergoing apoptosis because those are the ones that are dividing, perhaps. Or it could mean that the sphere is, again, resistant to this staining annexin 5 compound, and that basically it's unable to penetrate the sphere because of the diffusion gradient that Nikki mentioned earlier. So to try and clarify these two possibilities, you can actually use flow cytometry, where first you dissociate the sphere, and then you do your annexin stain. And if you saw the histogram on the top, where only a small number of cells stain green for annexin, that would indicate the first theory, where only those small portion of cells on the exterior are undergoing apoptosis and staining positive for annexin 5. If instead you see the histogram on the bottom, where a large portion of cells are stained green, this would indicate that the sphere is actually refractive to the annexin reagent. And so again, that can really help clarify that data. However, if you were only using flow cytometry alone and you saw the upper histogram where only a small portion of cells were positive, you wouldn't necessarily know that they were all on the exterior of the sphere without using the imaging data. So again, between these two analyses, you can get a very complete picture of what's going on. So I'll talk now about each of the forms of sample preparation that was required for these two different analyses. So after the 24-hour treatment, we stained the treated spheres with a variety of reagents, two of which I've already spoken of. But what we'll be showing from this point on is cells that were stained with cell event caspase 3,7 green, which is a reagent that in the presence of active caspase 3 or 7, the reagent is cleaved and then fluoresces green. And also propydium iodide, which is a very standard way of looking at cell membrane integrity. So if cells are undergoing cell death, they may become permeable to propydium iodide and glow red. And those two images are shown on the right, also in the presence of the nuke blue host 33342, which I mentioned earlier. Essentially for the sample prep, it was very straightforward. Much like the compound addition, all we did was replace 6 microliters of the media with 6 microliters of the staining reagent. And we then incubated the spheres for 1 to 4 hours, depending on, and basically imaged at multiple time points. Again, the automation goals here were to, 1, minimize the dead volume of our staining reagent, because some of these can be very expensive. Propydium iodide is not, but the caspase 3,7 was on the expensive side, so we certainly didn't want to waste our staining reagent. And because of this, we actually put them into a 96 well plate, much like we did for the compound dilution curves. We also wanted to avoid any lost wells due to pipetting or movement, and no lost spheres due to the removal of the media. And we also wanted to have the ability to image the sphere at 10x, and I'll talk a bit more about that at the end. So there's a lot to look at here, but we'll go through it a bit at a time. But basically, these are the same 48 wells that we showed earlier. Again, now having been stained for propydium iodide for the 3 columns on the left, or caspase for the 3 columns on the right, and showing 3 different compound treatments for 5-fluorouracil, camptothecin, and staurosporine. And again, as we know, the dilution curve moves down the column, so the maximal concentration's at the top. A 1 to 64 dilution is in the second to last row, and the last row contains a control DMSO-treated spheres. So the processing of a full plate was again accomplished in less than 8 minutes, so much like our compound treatment. What we do notice is that we were able to stain the interior cells. Again, basically the interior of the sphere are what seems to be glowing green or red as compared to the annexin 5 staining that we showed earlier. We did see a correlation between the caspase staining and the propydium iodide staining, which is nice in the sense that basically both conditions are a marker for apoptosis, and we do see nice correlation. And now we'll kind of walk through one treatment at a time from right to left. So in the staurosporine column, what we can see is a typical dose response, where the highest treatment concentration at the top gives you the brightest staining, and that amount of staining decreases as you move down the column as the compound was diluted. So pretty straightforward there. In contrast, camptothecin shows a very abnormal dose response curve, where our maximal effect was seen at the 1 to 4 to 1 to 8 dilutions, and that as you go up into the maximal or the 1 to 2 dilution, you actually see a decrease in the amount of fluorescence. And we saw this consistently, so this did seem to be a real phenomenon. Finally, 5-fluorouracil basically showed minimal, if any, effect, and certainly no dose response. So all we saw was kind of a low level of green or red staining, perhaps slightly higher than the control samples, but really nothing significant and nothing showing any sort of dose response. So we'll come back to that data a bit later, but I did want to touch on some additional lessons learned from the imaging process. One is that having water in the plate reservoirs was key. We initially tried some of the plates without the reservoirs, and what we did find is that evaporation was pretty significant, and what this would cause would be that the spheres would essentially ride up the droplet and not be in the field of focus. So once we added reservoir or water into the reservoirs to help mitigate that evaporation, things were very consistently in the plane of focus. However, we then basically did take Z-stacks to also improve our imaging. The Image Express has a great autofocus feature where it can find the bottom of the plate and then move a pre-assigned height, and for these hanging drops that was key. It would jump up to where the sphere should be located. But then to really kind of fine-tune in on that, we would take 30 images as a Z-stack and then basically take the one that was in the best field of focus. And we were able to accomplish this just using the bright field spheres, but we found that if we actually used the Hochstained channel, that would actually give us the best focus on the sphere. In addition, we found that incubation time was important, and that's to be expected, as Nikki touched on, where the diffusion gradient into spheres can be a significant factor. And there was even some differences based on what reagents you were using. And one of the other challenges, which we didn't necessarily anticipate when we began, was that some of the compounds we were treating the cells with were actually fluorescent. So for example, shown below is the highest concentration of nicotazole as well as being stained for propylium iodide, and what you can see compared to the control is significant red background there. So again, in the hanging drops, this is more challenging to remove than it would be in just a standard 2D culture. You could possibly do it through numerous wash and removal and replacement steps, but it would definitely be a significant challenge. And because we were doing a dilution curve, that background does change from well to well. So again, just another consideration for imaging. So flipping over to the flow cytometry sample prep, again, this is a very different sample prep. Obviously, the main focus here is to how we dissociated the spheroid into the single cell suspension. And basically, this process entailed transferring the spheres out of the perfecta plate and into a standard round bottom well plate. We then PBS rinsed the remaining media off of the cells and incubated the cells in Accumax, which is basically a mixture of trypsin and collagenase, a way of, again, chewing up the extracellular matrix. For 30 minutes, and then every 10 minutes of this period we would do a repeated pipetting step with our multi-channel head to dissociate the sphere further. Once the cells were dissociated, we added the staining reagents and incubated for 1 hour. Again, for our automation goals, we wanted to make sure that all spheres were transferred from the plate, none were left behind. We obviously wanted to make sure that we had complete sphere dissociation, as illustrated on the image on the lower right. Because without this, obviously, you can clog your cytometer or get uneven staining. And finally, because a number of flow cytometers actually require samples to be in tubes, including the gallios, we then had a final step where the samples were transferred into flow cytometry tubes from the plate. And again, we've got a video here to illustrate the automation. Shown here again is just the perfecta plate being delidded. And then we use the enhanced selective tips to take the first 2 columns worth of wells and basically mix them up and down to try and get the sphere into the pipette tips, and we dispense it into a plate with PBS that had been added by the span 8. And here you'll notice hopefully be able to see the pod move to the left. And again, that's kind of an illustration that shows the fine-tuned pipetting. We wanted to aspirate from the side of the well so that we didn't re-aspirate the spheroid as it sank to the bottom. We then used PBS to rinse out the wells, essentially. This would mainly only get the last 1 or 2 spheres that hadn't been taken up out of the entire plate, dispense it back, and then basically we would remove the PBS rinse and the residual media, again using enhanced selective tip pipetting. Again, also aspirating from the side of the well very slowly so as to not aspirate up the spheroid, dispense to waste, and then add the Accumax solution. The plate would then start shaking and shake for 30 minutes. We'll only show it for a few seconds here for demonstrations. And then every 10 minutes we would do 40 pipetting steps up and down, basically just trying to dissociate the sphere, which again is much easier to do in an automated 96 wells at a time fashion. This again shows the span 8 pipetters aspirating up the staining reagent from the reservoir, and now this actually shows you the span capabilities where we were able to dispense it to 4 tubes at a time, which you would not be able to do without the span capability. The span 8 then loaded fresh tips and aspirated up the dissociated cells from the plate, and again dispensed them into the tubes 4 at a time. Once all this was accomplished, those tubes were then incubated for 1 hour and analyzed on the Gallios flow cytometer. So shown here is basically the flow cytometry results. Again, we tried to put them in a very similar fashion to how we were showing the imaging results. So all of these wells correspond to the same wells you saw in the stained spheroid, with again propylium iodide on the left, caspase on the right, the three different treatments, and the concentration curve moving down the column. Again, here in this particular case, we've stained the positive portions or colored the positive portion of cells either red or green for illustrative purposes. So this full plate was processed in 48 minutes, again, mostly due to the incubation of 30-minute time. Again, we see a correlation between the PI staining and the caspase staining, indicating consistency across the 2 different processes. Starting again with the staurosporine, we see again a typical dose response where the highest concentration leads to the highest percentage of cells that are undergoing apoptosis. And again, the nice thing about flow cytometry is that we can actually quantitate this number, showing that roughly 70 or 66% of our cells stain positive for the various markers. And then as you move down the column, you'll see that red or green peak growing smaller and the gray peak growing larger, indicating the dose response. Again, consistent with what we saw with the imaging, the camptothecin maximal effect was seen in the 1:4 dilution. Again, also staining at around the 65 to 75% of our cells as positive for the marker for apoptosis. And again, what you can see up above is that the gray peak actually gets larger as you move up those concentrations. So it's not just that we're losing cells or that they've turned to debris. It actually appears that there's a larger portion of cells that are unaffected or at least not staying positive for these treatments. So again, very abnormal dose response curve. However, one of the most interesting effects is that we do see significant staining with our 5-fluorouracil treatment in this case. Upwards of 50 or more % of the cells staining positive for PI and caspase at our maximal concentration. And you can see a distinct typical dose response curve as that red peak or green peak gets smaller as you move down the column. So again, this is in direct contrast to what we saw with the imaging, indicating that, again, how these two different conditions two different analyses can be complementary. So what might be the case is that camptothecin and staurosporine are such harsh treatments that the cell-cell interactions or tight junctions may have been disrupted, allowing the staining reagents to have access to the inner cells. But 5-fluorouracil may not have done as much damage, and so again, you would get that diffusion gradient that would prohibit the staining. So again, just another instance of how having the two different analyses can be complementary. Some additional lessons learned from flow analysis is that we found that dissociation of the spheres became more challenging with either older spheres, those spheres that had been grown out to day 7, 8, or 9, were typically more difficult to dissociate, and also those that had the highest amount of cell death were the most difficult to dissociate, so typically our higher concentrations of compound treatment. Again, not to say they couldn't be dissociated, but those were the ones that required the most time or the most mixing. So those were sort of our bellwether wells as far as if we had sufficient dissociation. I've also shown how dose responses or how the two different analyses can differ between imaging and flow cytometry in the case of 5-fluorouracil. However, if we go back to the annexin image that I originally started this out with, we actually see a nice correlation where only a small portion of the cells were staining positive for green, indicating that, again, only those external cells seem to be staining positive for annexin. So finally, now that we've basically shown that we've been able to automate the plating, the treatment, and the analysis phase, how would one actually scale this up beyond just a single plate or even just a small portion of a plate? And for this, obviously, there are a number of considerations. One is you will have a higher throughput. So this does require that plate or tip storage that I mentioned earlier. And shown on the right in this image, again, this is just sort of a rendering of a potential screening system that does have the additional plate and tip storage that would allow what's shown here is the Biomek NXP with the enhanced selective tip multi-channel head, access to multiple plates that would then come in and out of the incubator shown here on the left, and eventually, after the processing has been completed, would be then placed using a pick-and-place robotics to directly put the plate into the high-content imagers such as the Image Express Micro. And by accomplishing this, this does allow you to automatically process an entire screen without any sort of intervention at any stage. From a hardware perspective, that's fine, but we also need software that is able to accommodate this. And one thing that is included would be optimizing the movement of labware to and from the liquid handler, and our software is capable of doing this. But also is capable of having a multi-day scheduling process. So again, all the 3 different steps that I mentioned are separated by multiple days, and our software can basically string those together and place them onto a calendar shown on the lower right, which allows you to get a feel for how much use the system is getting, if any of the steps require you to come in on the weekend or anything like that. So in addition, when it comes to data management, you can actually remotely monitor the system and see where it is in the process. Plus, any results you get from your analyzer can actually be used to drive additional pipetting steps, such as any hit picking steps or anything like that, if you wanted to actually take those certain wells forward for additional analysis. Also, you can even enter in data from an external analyzer if you're unable to have this complete automation system. So with that, I'd just like to wrap up. Hopefully, I've shown how we were able to successfully automate the plating, compound treatment, and sample prep of the perfected 3D plates and the spheroids that we were able to culture. I was certainly experienced that the greater control and consistency that comes from automation definitely was able to do a better job than I was manually, particularly when it came to transferring the spheres out of the HD plates, for example. I've also shown how the enhanced selective tip was able to accomplish additional things such as minimizing dead volume or doing row-wise dilutions. And I was able to show how integrations can certainly help you automate complete workflows, whether that's through integrating incubators or analyzers, etc. And finally, I hopefully showed you how there are certainly challenges for analyzing the cells in these 3D fashions, but how you can certainly complement high-content imaging with flow cytometry, how sometimes you'll get different answers, but they're answers you wouldn't necessarily get just using one or the other, and how imaging can really be valuable for getting sphere-wide data, while flow cytometry can be very valuable for getting cell-specific data. So with that, I'd just like to thank the people who are involved in this collaboration, in particular Nikki for her advice on using the plates and setting up the cell conditions. For molecular devices, Vipat Roxakultai was one of their application scientists who came and helped us with doing some of the initial imaging, finding the best conditions, and setting up the conditions to get all the great images that we had, as well as some of the analysis that was shown with the circularity and imaging in area. And finally, from the folks at Beckman Coulter, our integrated solutions group, who are basically assist with all the integrations on the Biomek, and our flow cytometry team, who's helpful, and also Chris Carpenter, who basically shot the videos that I showed today. So with that, thanks for your attention, and I'll turn it back over to Marianne for questions.
Thank you, Mike. And we do have a number of questions that have come in. The first one I'm going to address to you, Mike, and that is, there was a question whether you tried staining a spheroid and imaging it, then dissociating it and running it on the flow cytometer so that you had the same sample on two analysis platforms.
We did do that. Yeah, I didn't show that data, and that's a great question. And what we did find is that you would see what you would expect to see, essentially. So if you saw low staining on the imaging, you would get low staining on the flow cytometer. But essentially, that's still sort of asking the same question two different ways, as opposed to dissociating first and then staining, which if you did have the spheroid preventing the stain from attaching to the interior cells, you would not necessarily detect that difference. So FlowCyt, doing exactly what was suggested, did confirm that what you were seeing by imaging you could quantify slightly better with the flow cytometer, but you wouldn't necessarily see the differences that were due to prohibitory staining in the spheroids.
Okay, for the next question, I'm going to address to Nikki, and it concerns the use of using PVA to help form spheroids or other extracellular matrix. And one of the questions was the polyvinyl alcohol toxic to the cells? And another question was, how do I know how much to add?
Okay, so for the first part of that question, is polyvinyl alcohol toxic? No, if you check the literature, it's been used quite extensively in both cell biology and developmental biology for quite a long time to make hydrogels, essentially. So it's not really known to be toxic. And then for the question about knowing how much to add, this needs to be determined experimentally, depending on your cell type. And there's many ways to induce spheroid formation. You can use the packing agent like polyvinyl alcohol, or you can use extracellular matrix, matrigel, various components, collagen. And again, by utilizing these methods, you can make a very model-specific type of environment for the cells. We do have a protocol on our website that suggests some starting concentrations, but I really advise everyone to just set up some initial experiments and play with concentrations so they can derive the model that they're seeking to derive.
Yeah, just also I'll add to that. The reason we initially chose polyvinyl alcohol was we had done some earlier work using human iPS cells where without the PVA we couldn't get any sort of embryoid body, but in the presence of PVA we could. Again, for the HCT116 cells, it was not necessary. They would form excellent spheroids on their own. It was just really useful to kind of speed up the process.
Okay, Mike, and I have another question for you, and that was, what was the final DMSO concentration of the test compounds?
So that would have been essentially 0.25 microliters in per well. So basically we did a 1 to 100 dilution for the initial treatment, and then basically added the 10 to 40 microliters. So essentially 1 to 400 dilution of DMSO per well.
Okay, and Nikki, how stable are the drops in the hanging drop plates?
So I think that the videos that Mike showed of the robot moving the hanging drop plates does quite a bit to show how stable they are. There are two things that can grossly affect stability. First of all, sudden jarring movements are never good. Dropping the plates, banging them into things. You can imagine that this is just acceleration and deceleration that a hanging drop just can't handle. And the second thing that will sort of affect drop stability is exceeding the volumes. So for the 96 well plate, it's a 50 microliter max volume. If you exceed that, you'll find the drops becoming unstable. And then also, as Mike mentioned, obviously anything that would affect the surface tension of the media can affect it. But for the most part, they're really, really stable. It's no problem to pipette into them and pick them up and move them around, either with your hands or with a robot.
Okay, and this question maybe both of you could address, but I'll start with Mike because he went through a protocol. And the question is are there any protocol for 3D spheroids dissociation and preparation for flow cytometry available? Is the protocol you discussed available, Mike?
So again, yeah, I basically kind of determined what worked empirically, and I do think that's going to vary depending on the cell type you're using and some of the other factors that I mentioned. But what we used was basically a fairly straightforward way of just finding the best, Accumax is a pretty robust enzyme solution, and as far as how long it took to either incubate or the multiple pipetting up and down was basically just determined empirically. I'm happy to share that information with anything. Again, well, I guess I basically have, but for additional spheroids of either longer cultures or different cell types, we might have to change some of those conditions, and that would have to be determined empirically. Again, as far as the Biomek method or something like that, there's definitely ways we can get that out to people if they're interested as well. But basically, just from a standard protocol standpoint, it was really just time, temperature, and repeated pipetting, which is a lot easier using automation.
Nikki, do you have anything to add to that?
I would only add one thing, and this is people are going to have to determine this for their model for the most part, and I think that the key way to start is to try and figure out what extracellular matrix is being secreted by the cells that you're trying to split apart. As Mike alluded to earlier, Accumax is trypsin and dispace. If you have a lot of collagen that you've added, you might want to add a collagenase. And so that's the only thing I comment. It can be done probably for many different types of spheroids. It's just going to maybe take a little bit of thought and a couple experiments.
Okay, and Mike, could you address whether there are ways to quantify the imaging results?
Sure. Yep. And again, a big shout out to our folks at Molecular Devices who helped us with this. So again, what we looked at predominantly was, again, for the spheroid formation, we looked at area, perimeter, and shape factor. But for the fluorescence, for example, we did look at fluorescence intensity in the different channels. And so you could try and get an average, basically, of however intense your propydium iodide staining was, compare that to how intense your hoax staining was, and that would give you a rough idea of fluorescence per cell. But again, what it can't do, so that can give you a rough idea of sort of how bright on average the cells are. What it can't do is necessarily distinguish between having twice as many cells at the same intensity or having the same number of cells at twice the intensity. So it doesn't really give you a per cell, cell-by-cell data, but it can at least give you sort of an average fluorescence per cell in the entire sphere. So that was pretty much what we looked at. But there are certainly other ways to do additional analyses depending on what people are interested in.
Okay, we have time for one last question, and I'm going to address this one to Nikki. And Nikki, how long can spheroids be grown and analyzed in the hanging drop plates?
So we know of a customer who was able to grow spheroids for up to 40 days in the hanging drop plate. This is going to vary depending on how many cells you plate and the metabolic needs. Obviously, if you put a whole lot of cells into a small amount of media, you're going to have to change media more frequently, and it's going to have to be something you'll have to address with your model. And then I think that's the analysis part would go along with how long you can keep the spheroids alive and happy in the hanging drop plates. So as long as the cells are happy, you can analyze them.
Thank you, Nikki, and thank you, Mike. Well, this concludes today's webinar. If you do have additional questions, as I said earlier, the recorded session and the PDF of the presentation will be available on our website within 24 hours, and it will have both Nikki and Mike's contact information. Our next webinar, can you page to the next slide, Mike? Our next webinar, which is identification of novel stromal targets that support microtumor formation, will take place on February 18th. Our guest speaker will be Dr. Shane Horman of the Genomics Institute of the Novartis Research Foundation. And if you would like to, you can sign up for the webinar on our website. And if you would like to receive notices about our upcoming webinars, you can also sign up. So thank you for attending today. This concludes today's session.